Done. Okay. Hello guys. Today we are discussing the last part of our chapter, sexual reproduction in flowering plants. In the previous session, we have discussed about pollination and pollination devices and pistil pollen interaction. Today we are going to discuss the last part of our chapter, that means class 12 NCRT biology, that is sexual reproduction in flowering plant. Okay, before entering the chapter, do like, share and subscribe to our channel Mahatma Academy. Okay, let's start the lesson. Okay, let's start. Now the last part, it is okay, our last part is artificial hybridization. Now artificial hybridization means it is a crop improvement program in which desired pollen grains are used for pollination. That means it is carried out with the help of humans, so we can get the our particular products. Like if we want to improve the quantity of the rice, we can get the desired pollen grain, desired flowers and so on. Now the techniques used are emasculation and bagging. Okay. Now first we will see what is meant by emasculation. Emasculation means removal of anther from the bisexual flower bud of the female parent before the anther dehisces. That means in the bisexual flower the anther is removed. Now why they removed? If they are dehisced after some time, they will cause self-pollination. That's why they are removing. Now bagging. Here emasculated flowers are covered with a suitable bag made up of buttered paper. Okay, here that means the flowers which are removed with the anthers have covered with a suitable bag. What is the bag made up of? The bag is made up of butter paper to prevent contamination of the stigma with unwanted pollen. Okay, when the stigma attains receptivity, that means the stigma will attain receptivity means the correct pollen, the stigma can choose either it is correct pollen or not. Mature pollen grains collected from the anthers of the male parent are dusted on the stigma. Okay, mature pollen grains collected from the anthers of the male parent are dusted on the stigma. Then the flowers are rebagged. That means after removing the first bagging, they will do this step. That means and the anthers of the male parent are done on the mature pollen grains and then that dusted on the stigma. After that they can rebag and they develop the fruits. Now for unisexual flowers there is no need for emasculation. Why there is no need? Because it is there are no stigma and anthers on the same plant. Female flower buds are bagged before flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen grain and the flower rebagged. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flowers become rebagged. Now we are seeing about the double fertilization. After entering one of the synergids, look here. Here it is where the synergids. Yeah, here it is synergids. The pollen tube releases the two male gametes into the cytoplasm of the synergids. That means the pollen grain tube has two male gametes after development. Either after shedding two cell stage or having three cell stage, release the two male gametes into the cytoplasm of the synergid. One male gamete moves toward the cell that yes, it is a cell and fuses with the nucleus to form zygote. That is first one. The other male gamete go to here. Two polar nuclei. Okay, in the central cell and fuses with a triploid primary, and as it involves the fusion of three haploid nuclei or triple fusion. Okay, that means in the synergy they have nucleus. Look here, it forms a zygote here, and second one will form the fuse with to the two celled one. That means 1 plus 2 gives rise to 3. That is known as triple fusion. Since two types of fusion, that means syngamy and triple fusion, takes place in embryo sac, it is called double fertilization. It is an event unique to flowering plants. So it is an unique event to the flowering plant. The central cell after triple fusion becomes primary endosperm cell. Look here, it is the primary endosperm cell and develops into endosperm. Zygote develops into embryo. Okay, the central cell after triple fusion, that means the central cell will form primary endosperm cell here only and develops into an endosperm. Zygote develops into embryo. Now, what are the post-fertilization events? First, the endosperm will develop, embryo will develop, and ovaries will transform into seeds and ovary into the fruit. These are the one. Endosperm. That is polar nuclei, it is the egg. After that, it will form the sperm cells and it is zygote. After that, it will both will fuse into the endosperm. After that, the egg will form the zygote, and then it will form the embryo, whole embryo. Endosperm development. Primary endosperm cell divides repeatedly to form a triploid. That means one gives rise to 3. Okay. So look here. This primary endosperm cell will form 3 types of cells. Endosperm cells are filled with richer food materials. That means they help in the nutrition of the developing embryo. Look here. They are used for the nutrition of the developing embryo. That means they help in the nutrition for developing embryo. In common endosperm development, PEN undergoes successive nuclear divisions to give free nuclei. That means in common endosperm development, primary endosperm nuclei undergoes successive nuclear divisions to give free nuclei. Okay, it will form the new free nuclei. That cell is called free nuclear endosperm. Number of nuclei, free nuclei varies greatly. Look, this is the free nuclei. This is one. Central cell. Endosperm becomes cellular due to cell wall formation. Here cell will form, that is called cellular formation. Endosperm development. Tender coconut water is a free nuclear endosperm. That means it is free of nucleus. That is central cell. So made up of thousands of nuclei. And the surrounding white kernel is the cellular endosperm. That means this is a white kernel, that is cellular endosperm, and this is the last thousands of nuclei, coconut water. Now, embryo development. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. This is the embryo, this is the micropylar end. Embryo will develop at the place where the zygote develops. Most zygotes divide only after the formation of some endosperm. Okay? Most of it will divide when only after the formation of some endosperm. This gives nutrition to the developing embryo. That means what is the nutrition for the developing embryo? Endosperm. In monocots and dicots, seeds differ greatly. Okay? In what seeds differ greatly? In monocots and dicots. But embryogeny, that means early embryonic development, is similar. Look here, these are the steps. Zygote, pro-embryo, globular, heart-shaped embryo, and mature embryo. This is the zygote, this is the pro-embryo, this is the globular, look, it is like globe, and it is heart-shaped. With the mature embryo, this is the one. Now, we are saying about the dicotyledonous embryo. It has embryo it has an embryonal axis. Okay, it has an embryonal axis and two cotyledons. That means cotyledons means two cotyledons means dicot. One cotyledon means monocot. The level of embryonal axis above the level of cotyledon is the epicotyle. That means above. That means epi. Downward means hypocotyle, which terminates with the pumule. That means epicotyle will form the pumule, that is stem part, and hypocotyle will form the root tip. The root tip is covered by the root cap, but root cap is absent in hydrophytes. Now we are seeing about the monocotyledonus. They possess only one cotyledon. Cotyledon of the grass family is called scutellum. It is situated lateral to the embryonal axis. That means it is situated way to the embryonal axis. It is situated lateral to the embryonal axis. At the lower end, lower means radical, and the root cap includes coleorhiza. That means an undifferentiated sheath. Look here. Lower end means radical, upper end means epicotyle. Epicotyle means pumule. You can say. That means coleoptile. Lower means coleorhiza. Now seed from embryo. How seed from ovule will form? Seed is a fertilized ovule inside the fruits. Okay. Seed is what? Seed is a fertilized ovule formed inside the fruits. It is the final product of sexual reproduction. So why sexual reproduction will occur? To form the new embryos. That means seed is the final product of the sexual reproduction. It consists of what? Seed coat, cotyledons, and embryonal axis. This is the seed coat. Seed coat means outer layer. Cotyledons, if it is monocot, only one cotyledon. So this is a cotyledon. And if it is dicot, it has two cotyledons. Embryonal axis. Upper part means pumule and lower part is radical. Upper means stem, lower means root. The cotyledon is simple, genetic and swollen due to shortage of food. That means they store food. What is the storage part of the seed? It is endosperm. Endosperm provides nutrition for the developing embryo. Metastases are of two types, non-albuminous and albuminous. Non-albuminous means they have no endosperm, while albuminous means they have complete endosperm and is completely consumed during embryo development. During embryo development, what is completely consumed? Endosperm. What are the examples? Peas, groundnut and beans. Examples of non-albuminous seeds are pea, groundnut and beans. And albuminous ones, wheat, maize, barley, castor and coconut and sunflower. Same mature seeds. Here, mature seeds. But you can see the diagram is not different. There are two cotyledons and a micropyle, hypocotyle. Occasionally in some seeds like black pepper, beet, etc., remnants of nucleus are also persistent. It goes perisperm. That means in some plants, in some seeds like black pepper and beet, there are some examples. There are examples of which remnants of nucleus are also persistent. So perisperm. Here, where is the perisperm? Okay, pericarp. These are the examples. Integuments of ovules hardened as tough protective seed coats. That means it forms a tough protective seed coats. What form the tough protective seed coats? Integuments. There is a small pore through which O2 and water enter into the seed during germination. During germination, what enters into the seed or the ovule? Oxygen and water, through which they will enter micropyle. As seed matures, it becomes dry by reducing water content. Okay? As they mature, you can feel that the seed will not have any water content, but it has water content. That the water content is 10 to 50% moisture by mass. The metabolic activity of the embryo slows down. Why the slows down? Because the favorable condition, that means enzyme will require water for their activity. So they development and metabolic activity will slow down. It may enter a stage of inactivity. Inactivity means dormant. It is during unfavorable seasons. During favorable seasons, that is moisture, wood, and suitable climate, they hibernate. I mean, during favorable conditions, what they provide? They provide moisture, oxygen, and suitable temperature. Now we will see what are the advantages. Since pollination and fertilization are independent of water, seed formation is more dependable. That means pollination and fertilization do not require water, so seed formation is more dependable. Better adaptive strategies for dispersal to new habits. That means if in condition there are unfavorable conditions, so in the foreign place there are favorable conditions, so the seeds will travel from unfavorable conditions to unfavorable conditions. It helps the species to colonize in other species. Colonization and the seed formation is more dependable. They have food reserves. That means they have endosperm. That is why they can nourish the developing embryo. So young seedlings are nourished until they are capable for photosynthesis. And the hard seed coat. They have seed coat. They protect the seed from the from the stress and external conditions. Okay, being product of sexual reproduction, they generate new genetic variations and combinations. Okay, that means they are produced from the two different parents, so they show genetic variations and combinations. Dehydration and dormancy of messages are crucial for the storage of food seeds. That means dehydration, they lose water, and dormancy, that means they are in the stage of inactive growth, inactive metabolic activity, and so on. It can be used as a food throughout the year and also to rise the crop in the next season. That means due to these properties, that means dehydration and dormancy, they can use as food throughout the year and also to rise the crop in the next season. Now, I think this is the last concept. Okay, we have still more. In a few species, the seed load viability within a few months. That means viability means the lifetime of the seeds. That means in a few species, the seeds lose viability in a few months, while some live for many years. Some seeds can remain alive for thousands of years. That means they can live for thousands of years. Also, the oldest is that of a lupine. That means Lupinus arcticus. It is the oldest seed. Excavated from the Arctic tundra. Okay, it is excavated from the Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. So it has what the dormancy of this Lupinus arcticus. It has 10,000 years. That means the seed germinated after 10,000 years of dormancy. It is germinated and flowered. And it's a date plum. It has 2,000 years of old viable seed. Okay, this is the one. Now we are seeing the fruit from embryo. The ovary develops into a fruit, ovule into seed. The transformation of ovules into seed and proceeds simultaneously. That means if the fruit, that means seed converts into ovule and the seeds over into fruit, it will occur simultaneously. The wall of ovary develops into pericarp. That means ovary wall develops into pericarp, that means outer layer. The fruit means fleshy or dry, depending on the type. What is the fleshy? Guava, orange, and mango. What is the dry? Groundnut and mustard. once again, fleshy one are guava, orange and mango, and the dry one are groundnut and mustard. There are two types, that means true fruits, false fruits. First, true fruits. True fruits are developed from the ovary, while false fruits are developed apart from the ovary. That means they are developed from thalamus, stigma, and so on. Okay? What are the examples of the true fruits? Most plants. And the false fruits are apple, strawberry, and cashew. Okay? Look here, it is apple, strawberry and cashew nut. Now, in some plants, fruits can develop without fertilization. Such fruits are called parthenocarpic plants. Example, banana. That means the banana, as you see, banana do not have any seeds. Why they do not have any seeds? Because it does not develop with the help of fertilization. So, no seeds and so on. That is why banana is called parthenocarpic fruits. Parthenocarpic can be induced through the application of growth hormones. How this occurs? It occurs with the help of the growth hormones. Such fruits are called seedless fruits. And parthenogenesis means development of fruits with the help of the without the help of fertilization. Apomixis means single. So, production of seeds without fertilization. What are the examples? Asteraceae and grasses. It is the form of asexual reproduction that mimics sexual reproduction. That means it is a asexual reproduction. Look here, apo means single. Amphi means sexual. That means double. That mimics a sexual reproduction. Development of apomytic seeds. In some species, diploid egg cell is formed without reduction division and develop into embryo without fertilization. That means there are some species in which diploid egg cell will convert into the embryo without fertilization and without reduction division. So it involves two steps. That means it skips two steps. One is reduction division and one is fertilization. In many species, that means citrus and mango, some nuclear cells surrounding the embryo sac divide, protrude into embryo sac to form embryos. That means nuclear cells. Nuclear cell is present in which one? Embryo sac. It produces into the embryo sac to form embryos. So, embryo sac will develop into embryo. Through which process? Nuclear cells will divide and it produces into embryo sac. Thus, each ovule contains many ovaries. That is why each ovule has how many embryos? Many. Occurrence of more than one embryo is called, in a seed is called polyembryony. So, we have completed apomixis and polyembryony. Now, we are seeing the importance. If the seed from hybrids are sown, the plants in the progeny separate and lose hybrid characters. That means if we collect a plant that is hybrid and from that we collect the seeds, and that the plants in the progeny will segregate, that means divide and lose hybrid characters. Production of hybrid seeds is costly, so hybrid seeds are expensive. That is why we can see that hybrid seeds are very expensive. If the hybrids are made into apomix, there is no segregation of character in the hybrid progeny. So farmers can keep on using hybrid seeds to raise new crop. So first, when the hybrid cells are sown, the plant will in the progeny, that means embryo, will segregate and it will lose characters. That defects are costly and they're expensive. Now, if they made into apomix, there is no segregation. So the farmer can easily keep on using hybrids to raise snake crop. Okay guys, we have completed the sexual reproduction in flowering plants chapter. In the next session we will start with the human reproduction chapter. Until then, take care and bye bye. At last, don't forget to subscribe to our channel Mahatma Academy. Bye bye.